This video is about evidence for evolution, evolution in action, and imperfections in evolution. So, DNA evidence is the genetic code. They are the triplets of DNA nitrogen base sequence that code for specific amino acids. The amino acid triplet is the same in almost all organisms. The similarity of triplet DNA codes making up amino acid shows a probable common ancestor for all life on Earth. So, this shows genetic code. Universal genetic code. Similar genes over the ages, the genetic code has passed unexchanged or nearly so from parent to offspring. Amino acid sequencing and amino acid sequencing in a particular protein is compared between organisms. Comparing amino acid sequence shows amino acid sequence probably the strongest evidence for relationship among organisms. Number of differences for human hemoglobin. Then cladogram that shows the evolutionary relationship among group of organisms. Where would the common ancestor be? Number of amino acid difference in hemoglobin compared with human. In shortly, DNA sequence comparison is highly successful technique employed to study phylogenetic tree in instances where other evidences are scarce. For example, neutral human DNA sequences are approximately 1.2% divergent from gorilla and 6.6% divergent from baboons. Sequence evidence does enable quantification of genetic relatedness between humans and other apes. A classic example of biochemical evidence for evolution is a variance of cytochrome C in living cells. It is a respiratory pigment associated with electron transport chain of all eukaryotes. It is formed of 104 amino acid residues. The sequence of these amino acids varies with species. The presence of cytochrome C invariably in all eukaryotes reveals the common ancestry of eukaryotes. At the same time, its variation in different species probably indicate evolutionary changes in the different species. The cytochrome C molecule of chimpanzees and human beings are identical, clearly indicate that the two groups share a close common ancestry. And embryological evidence. Embryology is an important source of evidence for evolution. Every sexually reproducible animal starts its life from a single cell of the zygote. The zygote develops first into blastula, then finally to an organism. The development history of an individual organism is called the ontogeny. It is strikingly similar in related species at the different groups of vertebrates. Embryos of different vertebrate groups are also very much similar in the external form and structure. This makes them rather indistinguishable from each other. This striking similarities in the embryonic stages of different vertebrate groups are the following: similar form and structure of embryo with gill clefts, notochord, tail, and rudiments of eyes and ears, subsequent replacement of notochord by vertebral column, development of limbs from limb buds and tetrapods. The common pattern of development and striking similarities between the embryo of different vertebrates group clearly indicate that all vertebrates share a common ancestry. The biogenetic law. It was proposed by German biologist Fritz Müller and Ernst Haeckel, which holds that ontogeny is a short recapitulation of phylogeny. Ontogeny is the mechanical course of phylogeny is the mechanical course of ontogeny. This means developmental history of an animal is a brief recapitulation of the evolutionary ancestry of its race. According to the biogenetic law, and organisms during its early development briefly repeats or recreates the evolutionary stages of a group. In other words, it climbs up with family tree by passing through the stages which resemble its adult ancestors. An example is the appearance of gill slits in the embryos of lung-breathing vertebrates, reptiles, birds, and mammals. This reveals that terrestrial vertebrates might have evolved from gill-breathing aquatic ancestors, namely fishes. Though Haeckel's biogenetic law was decredited later on, embryology still provides several other evidences for organic evolution. Barnacles, for instance, are sedentary crustaceans with little apparent similarity with other crustaceans such as lobsters, shrimps, copepods. Yet the barnacles pass through the free swimming larval stage, which is similar to other crustacean larvae. The similarity of larval stages supports the conclusion that the crustaceans have a common ancestry. Similarly, a wide variety of organisms have very similar sequence of genes that are active during early development. These genes influence the same traits in all other diverse groups. The presence of similar genes controlling the same traits in wide range of organisms very well explain their presence in a very early common ancestor for this group. Human, monkey, pig, chicken, salamander, phylogeny, and ontogeny. This shows recapitulation theory. Fossil evidence or paleontological evidence. Fossils are the remains of organisms of a bygone ages remaining embedded in a strata on rocks. Since there are the actual remains of organism, they provide direct and most reliable evidence in favor of evolution and furnish detailed information about and ancestry of organisms. These fossils are extremely important for understanding the evolutionary history of life on Earth. The study of fossils revealed the form, structure, mode of life, behavior, interrelations, and evolution history of extinct organisms. The study of fossil records bring to light the following effect in support of organic evolution. Organisms of present day are very much different from those of distant past. In course of time, simple and primitive organisms and undergo transformation and give rise complex and advanced forms. Organisms which flourish at one time and experience each at a later time, giving way to the more successful forms. Mammals and angiosperm plants are the last revolt group. Skeletal structures or other hard parts of organisms are the commonest fossilization remainings. Petrification. If the animal gets subsequently buried in mud, mineral salts will infiltrate into its bones and fill their pores. As a result, bone will harden and get preserved as fossil. This process is called petrification. It is possible to find out how a particular group of organisms evolved by arranging fossil records into a chronological sequence. The lowest stratum containing the oldest rock and the earliest fossil, while the uppermost stratum contains the youngest rock and the latest fossil. Studies on the forces at different stratigraphic levels are shown that revolutionary changes have a coding time, increasing the variety and complexity of organisms. Some fossils. Then geological evidence, biogeography, divergent evolution. After the supercontinent Pangaea separated, several species diverged due to different environments. The geographic distribution of organisms on the planet follows patterns that are best explained by evolution and conjugation with the movement of tectonic plates over geological time. Broad groups that evolved before the breakup of the supercontinent Pangaea about 200 million years ago are contributed worldwide. Groups that evolved since the breakup appear uniquely in regions of the planet. For example, the unique flora and fauna of the northern continents are formed from the supercontinent Laurasia, and the southernest continents are formed from the supercontinent Gondwana. The presence of proteins in Australia, Southern Africa, and South America are best explained by the plant family Spiracines and the repair of the southern supercontinent Gondwana breaking up. Biogeography, convergent evolution. Unrelated species from different regions around the world converge to begin to look alike when found in similar environments. Giant armadillo, giant anteater, giant pangolin, spiny anteater. North America, Africa, South America, and Oceania. Divergent evolution, parent species. Convergent evolution, parent species, parent species. Then parallel evolution, parent species. Evolution in action. Evolution acting over very long time scales, billions of years, can give rise to diverse life forms and biological complexity, but it is possible to directly observe evolution has been in action. The three different areas where evolution has been observed in action. Evolution in action. Plants resistant to herbicides. Modern herbicides make major contributions to global food products by easily removing weeds and substituting for destructive soil cultivation. However, persistent herbicide selection of huge weed numbers across vast areas can result in the rapid evolution of herbicide resistance. Herbicides target specific enzymes and mutations are selected that confer resistance in doing amino acid substitutions, decreasing herbicide binding. So, where herbicides bind within an enzyme catalytic site, very few mutations are resistant while conserving enzyme functionality. Where herbicides bind away from a catalytic site, many resistant endoing mutations may evolve. Increasingly, resistance evolves due to mechanisms limiting herbicide reaching target sites, especially threatening are herbicide degrading cytochrome P415 enzymes able to detoxify existing new and even herbicides yet to be discovered. Global weed species are accumulating resistance mechanisms, displaying multiple resistance across many herbicides and posing a great challenge to herbicide substantiability in world agriculture. Fascinating genetic issues associated with resistance evolution remain to be investigated, especially the possibility of herbicide stress unleashing epigenetic gene expression. Understanding resistance and building substantiable solution to herbicide resistance evolution are necessary and worthy challenges. Then rapid evolution in the wild. Adaptation to an environmental change requires that the population already has a mutant individual who can thrive in the new environment, or that such a mutant arise before the population goes extinct. This is not always the case, especially when the environment shifts too much or too quickly, or when populations are too small and lack genetic variation. The race between adaptation and extinction is taking place for millions of species at a very moment. One example is provided by the Tasmanian devil, whose population has shrunk by 80% in just 20 years due to contact between individuals. In recent years, however, mutations that increase immunity towards the cancer have also spread in the population, with the endangered Tasmanian devil adapt to become resistant to this cancer and escape extinction is being a question for the scientific community. When drought struck the Galapagos Island, it severely reduced the supply of the small seeds eaten by the medium ground finch, a bird species living in the island. The birds had to turn to large seeds which could only be cracked open by individuals with bigger beaks. This gave a strong advantage to large beaked birds who could survive the drought and pass on their genes to the next generation. As a result, the average beak size increased in the just 2 generations. An evolution in lab. Studying bacterial evolution in the lab is also given scientists vital clues about the bacteria can accumulate genetic mutations that make them resistant to antibiotics. Understanding and combating antibiotic resistance is one of the most pressing scientific and medical challenges in the world today. We will dive into the fascinating world of bacteria and their ability to outsmart modern medicine in a later issue. Due to their short generation time and fast reproduction, we can select and evolve microorganisms that do many useful things for us, from strains of yeast that produce the best beer to bacteria that can break up toxic chemicals in our wastewater to microorganisms that are extremely efficient at producing biofuels. There is even a strain of bacteria that can collect gold from seawater. Some evolution examples of species and their changes over many generations. Peppered moth. The light-colored moth became darker after the industrial evolution due to the pollution of the time. The mutation allowed dark peppered moths to blend into their sooty surroundings and avoid being eaten by predators. Soon after, peppered moths were black instead of light, but notably the lighter color returned to population when pollution declined. Then pesticide-resistant insects. Whenever you use a pesticide, it kills a majority of the insect population. However, certain insects will experience a gene mutation to develop immunity to it, and those insects will reproduce. This happens very quick within a few generations, since the generation length of insects is short. Then bright colored peacocks. Females always choose the male peacock with the brightest color feathers. The genes for bright colors are passed down to male offspring, who also have a better chance for finding a mate and passing down their genes. The Darwin finches, the finches on the Galapagos Island, have all developed different beaks. Originally, the finches had large beaks for cracking large nuts. A group of finches come from another island who were larger and drove those away and ate their nuts. Over time, the finches developed beaks to eat smaller nuts that other finches did not eat. Evolution in coronavirus spike. Evidence for a common evolutionary origin of coronavirus spike protein receptor binding subunits. Among different coronavirus genera, the receptor binding S1 proteins, subunits of their spike proteins, differ in primary, secondary, and tertiary structure. This study identified shared structural topologies, connectivity of secondary structural elements in S1 domains of different coronavirus genera. This results suggest that coronavirus S1 subunits share a common evolutionary origin but have shared diverse sequences and structures following extensive divergent evolution. The results also increase understanding of the structure and function of coronavirus S1 domain, whose tertiary structure are currently unknown. Imperfections in evolution. Most of the time, evolution seems to do a pretty good job of turning out animals with adaptations that help them survive and thrive. But sometimes it doesn't entirely seem to make sense. One misconception about natural selection is that over time, Evolution selects the features of an organism that are most perfectly suited to its environment. This misunderstanding may be partly due to the term natural selection itself, which conjures up parallels with, say, dog breeders selecting for desirable traits in their animals. In fact, nature isn't always selecting anything. Natural selection is a process, not a conscious force. Selecting traits for dog breeds is a very different process to natural selection. There are good reasons why the process of natural selection may not always result in a perfect solution. Firstly, selection can only act on the available genetic variation. A cheetah, for example, can't evolve to run faster if there is no faster gene variant available. Secondly, the body has to work with the materials it already has. It can't make something out of nothing. That's why winged horses are the stuff of myth. Evolution also has to work with the developmental patterns established in distant ancestors, and the results sometimes seem very strange. For instance, I think it would make most sense for the nerve that goes from voice box to the brain in the giraffe to make the most direct route length of around 10 centimeters. But because the giraffe's body plan was established in the ancestor but had no neck, the nerve goes all the way down to the neck, around the heart, and back again a distance from few. Evolution also has to work with the developmental patterns established in distant ancestors. The results sometimes seem very strange. For instance, it would make most sense for the nerve that goes from the voice box to the brain and the giraffe to make the most direct route. But because the giraffe's body plan was established in an ancestor that had no neck, the nerve goes all the way down the neck, around the heart, and back again, a distance of 4 meters. Sending a nerve impulse from brain to voice box via the recurrent laryngeal nerve and vagus nerve in a giraffe involves a lengthy jitter past of the heart.